get started just to kind of get sure that we will be on time. Right now, 10 a.m. Eastern time. Thank you everyone to join. We have people from China that it would be something like midnight for them. We have people from India, from Iran, from different European countries. And we also have people in the kind of middle of the US that will be something like 8 a.m. for them. So we try to kind of start the meeting in a time that would work kind of as much as possible for everyone across the world. And it's great to have people. Just to start, I think that it would be great if Marom starts as one of the leaders in the field, discussing about the ideas that we have been sharing together about potentials for a collaborative network. Marom, please.
Thanks. Well, really, Hamed's the one. But I just want to say I feel so. It's first of all, it's really wonderful that everyone is here on this call. And everyone's like, especially people in weird time zones. It just shows that there's a general interest. And I just want to say I feel very strongly that without a group effort like this, without an international organization, we may not be successful. I mean, everyone here is interested in non-invasive neuromodulation for addiction. Everyone here is published on the topic. And just my experience from seeing what happened with depression, seeing what happened with other indications, I just feel very strongly that this effort is basically needed, or else we're going to be having the same discussion in 10 years, right? Well, if we don't come together and organize and focus, we will literally have the same discussion in 10 years.
Yep, that's true. And hopefully it would be a good starting point in a critical time period that people are getting more and more interested to the field, and they are hopeful to be able to kind of do really good things. Anna, good morning. I mean, good afternoon. How are you doing?
Hi, this is Anneke from Amsterdam, from the Netherlands. Good afternoon. Yeah, it's 4 p.m. here.
Oh, good. Great.
Wonderful to see everyone.
Great. So we have something like 5 minutes for introduction. I mean, if there is anybody who likes to kind of do any introduction, that would be nice. We can have kind of discussions and just simple introduction. So Anna, do you want to start to discuss about kind of introduce yourself and see how we might be able to move forward?
Hi. Yes, well, I think first thanks, Hamed, for pulling this together. I think it's very much needed in a field which is developing, and you could say that TMS in addiction is still in its infancy. I have been doing, so in our lab, we have been doing like an acute rTMS trial in alcohol dependence. And we started that a couple of years ago, and it has been finished already. And now we're doing a clinical trial focusing on high-frequency DLPFC stimulation in alcohol-dependent patients. And we're very interested in also investigating the effects on neurocognitive functions like impulsivity and executive functions, and effects on we're also combining it with a functional MRI study before and after.
Great. So yeah, we will have kind of half of the meeting discussing about things that people are doing right now in the field, and Anna would provide us with kind of few slides and discussing about kind of what she is doing in Netherlands. But just really short introductions for just kind of introducing yourself and where you are from, and then we would move forward for the next steps. Martin, do you want to
I'm sorry.
Martin, do you want to go ahead?
Yes, thank you. Do you understand me? That's
yes.
Me. I'm located in Würzburg in the psychiatric department. We started some years ago with tDCS and TMS studies, and the last 2 years we have some renovation in the building, so we focused on anxiety disorders and could not investigate addiction. But now we opened again the ward in which we have alcohol-dependent patients, so I'm happy to join this collaboration. And I prepared some slides, but this will be 1 hour later, isn't it?
Yeah, that's true.
That's all.
Great.
And my colleague sitting here 5 minutes ago was Thomas Polak. He's doing the work together with me.
Great.
Thank you.
Great. Grazia, do you want to go ahead?
Would you like some coffee?
Yes, of course. Me, right?
Yes.
I'm here to represent Professor D'Alimberti and Professor Bonci. We've been collaborating with the National Institute of Health, and we've been doing this study focusing on cocaine patients undergoing to rTMS on the PFC, and we are focusing on the effects of TMS on long-term follow-up. We have more than 3 years of follow-up for our patients, and we are also combining neuroimaging to our protocol of TMS. In addition, we are also starting a trial on gambling patients, and so we are focusing also on this area of behavioral addiction.
Great. Greg, would you move forward?
Oh, sure. Hi. Yeah, I'm Greg Salem. I work over at Medical University of South Carolina. As some of you may know, there's a fairly large group of us that are interested in this area, and we're doing several studies. The 2 studies I'm specifically working on is I'm on a Q23 right now, and the project for my Q23 is to try to use just a pretty standard excitatory rTMS to the DLPFC in cannabis use disorder. We're also just about to launch a study where we'll be trying to use the same similar stimulation patterns in people who are admitted for detox of opiate use disorder. So that should start maybe even just this week. I'm not sure if others from MUSC are on the call or not, but we're kind of working with every different population, and we're trying a couple of different strategies. There's a lot of excitement over here in this area. So thanks so much for organizing this and for everyone being here.
Great. Yeah, we have been kind of planning to have Colleen Hanlon with us today, but she had a grand round in Johns Hopkins, so she would not be able to join us, but she conveys kind of her greetings, and she is kind of one of the leaders in the group. So we are really happy that we have kind of really good people from MUSC. Zhao Chu, would you go ahead?
Okay. Actually, I'm come from China. I think come from the University of Science and Technology of China, in the Life Science School, because this is not a hotel. So I'm working on the neural basis of the. Actually, we're working on the tDCS and tES. I'm not working on the TMS. So we work on what the neural basis is of the tDCS effect, how to regulate the craving for the cigarette, for some heroin addiction drugs. We use fMRI and the EEG, and another thing is we try to build some new system, new tDCS system, a new tES. For example, we try to build the EEG guide the tDCS system. For example, we try to increase the simulation window for the tCS or tDCS for some issues. Another thing is we try to build some new machines to transfer. For example, do you remember the last year there one paper published in Cell? We can try to simulate the green area inside the make sense. So we try to build system tCS. Okay, that's all. Thank you.
Great. Christos from London, would you introduce yourself?
Oh, hello guys. Yes, I'm a consultant psychiatrist here in London. in addictions. I work in the NHS, the National Health System here. I'm also collaborating with Imperial College. But my relation with RTMS is through my private practice, the London Psychiatry Centre. We have introduced RTMS in the UK, and that is going on for 6 years now, treating people with depression, anxiety, fibromyalgia, pain. I haven't used it in addictions. It's a private setup, so it is quite expensive, and in the private setting. So I'm just keen to see where you are from a research point of view and how we could collaborate in the future.
Great. So sounds great. So just try to be on time for the next steps. You will have time at the end of the webinar to discuss and introduce other colleagues that are going to present their works, and also we will have time to discuss about potentials for extending the collaboration. The next step, I'm going to give a kind of a short presentation about where we are. So for the next 15-20 minutes, I will try to give you an idea about what are the things that we are kind of doing in the field. So in terms of how is the current status of tES TMS trials in the field, can you see my screen right now? Everyone could see my screen?
Yes.
Good. So good. Okay.
Yeah.
Great.
Yes.
So, like many other kind of labs, we have published kind of in different fields of brain stimulation and addiction medicine. But I'm not going to discuss about what we are doing right now and what we have published in the field. What I'm trying to do today is just to give you an idea about what are the things that are happening in the field and how many published studies we have in the field of addiction medicine. I recently sent you a database that we have recently collected with a systematic review in terms of seeing how many published studies we already have in the field. I mean, how many published trials we have in the field. We collected 34 tDCS trials and 50 RTMS trials in the field. So we already have, based on the search strategy that we have 50 trials in TMS and 34 in tDCS. And I shared the database with you guys, so it would be great if you take a look inside and see if you have missed any specific kind of original publication in the field that should be added to the database. But based on this database that we have, in terms of how different countries are being involved in the field, how many publications that we have from different countries, you can see that we have contribution from many different countries. So as you can imagine, that something like just 20% of the publications in the field are from US. And something like 80% of the publications come from different countries, from Austria, Belgium, Brazil, Canada, China, Czech, France, Germany. So we can see that we have contributions from people across the world. And then when we go to the details in terms of how people from different countries are contributing to the process, you can see that in terms of tDCS versus TMS, we have countries that are more active with TMS like Italy and countries that are more active with tDCS like Brazil. So you can see how different countries are contributing to the current pool of published articles in the field. But in terms of different types of drugs that people are targeting in their studies across the world, so that is the overview. And if you want to kind of make a guess about colors, because I've removed the color codes, what do you think about the green color? What is the green color? What type of drug would be green color here in this graph? What would be orange?
Alcohol.
Orange alcohol?
Or cocaine?
Cocaine.
Green is cocaine.
Cocaine. What about green is cocaine?
No, it's tobacco.
Green is tobacco.
Orange is
Orange is
What
cocaine.
about light blue? What would be
Alcohol.
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 It's the blue.
Okay, this is the color code. Right. Nicotine is the green, and blue,
orange
right,
is
kind of
cocaine.
light blue is alcohol, and yeah, cocaine is
Orange.
yeah, orange. So you can see that different countries are contributing to different, based on their national interests and what they face in terms of the problems with different drugs. So you can see different countries are contributing to kind of different types of drugs. And then in terms of how different drugs receive attention from different methods in terms of tDCS versus TMS, you can see that, as you can imagine, people are using both tDCS and TMS for almost all types of drugs that we have in the field. And but meanwhile, you can see that there are drugs that they have received less attention in the field. And then in terms of the registered trial in clinicaltrial.gov, you can see that we have ongoing clinical trials across the world that are right now registered in clinicaltrial.gov. And you know that clinicaltrial.gov is not the only registry for ongoing clinical trials. There is another large registry by WHO. But meanwhile, you can see that how different countries are contributing with ongoing clinical trials in tDCS and also in TMS. So you can see that different countries across the world that are contributing to ongoing clinical trials. But in terms of the numbers of the subjects that we have in the field, and numbers of the subjects in each arm of different trial, I'm sure that people have seen this publication, this recent publication, in terms of importance of sample size. The bottom line of this publication would go for just mentioning that when we have sample size of less than, let's say, 50 subjects in the trial, in the parallel design trial, something like 35 subjects in each arm, then the likelihood of finding an effect in this scenario that they have done was something like less than 80%. And then with a kind of small sample size, when people just find one positive significant test and report it, they always report a larger effect size. So with the smaller trials, we have larger effect size and lots of false positives there. So we know that we have, when we have a smaller sample size, especially in the field of brain stimulation, it seems that we would have significant problems. And in terms of what we already have in the field, in terms of the size of the trials, as you can see here right now, most of the studies in the field, they have less than 35 subjects in each arm. So we know that most of the published trials in the field, they suffer from having enough sample size to claim an effect. So it's really kind of a really complex issue in terms of kind of the how we might be able to rely on the publishers. And we are, I mean, us, like many other labs, that are guilty in the kind of the same issue. We have been exploring before, but it seems that right now we need to move forward for kind of larger clinical trials in the next steps. And then in terms of areas to target, we know that people are targeting different areas. Most of the people are targeting left DLPFC, but there are many studies with kind of right DLPFC, and also many other areas that people are exploring. So there are still kind of many questions about what are the areas that should be targeted. And we all know that there are different potentials. We have published about that in terms of what are the different potential montages or sites for even tDCS. And then we recently published a new kind of trial showing that these different montages would have different effects. But we can imagine that they can have different effects on different types of cognitive functions. So there are lots of challenges for that as well. And then in terms of the numbers of the sessions that people are doing the brain stimulation, So as you can imagine, most of the studies are just one single session of stimulation, and there are just few studies that they have kind of multiple sessions of stimulation. And then in terms of the follow-up, most of the studies, they do not have any follow-up. So something like 70% of the studies, they do not have any follow-up. And that would be another challenge in the field. And then in terms of the stimulation, when to stimulate. So there are many studies that people have tried to do a stimulation among those who are still not treatment seekers, or those who are treatment seekers, or those who are in the early stage of recovery in the first few days in the detoxification, or those who are in the recovery. And how our brain stimulation protocol is going to interact with ongoing interventions and treatment is another challenge that we should kind of handle in the future. And if we kind of see this graph in terms of different drugs, you can see that almost all nicotine studies have been done before starting the treatment. So they are pre-treatment trials. And as you can see that there would be different kind of, for example, those who have done the studies with cocaine, they have done studies in different levels of recovery, and how these things are going to interact, that would be another question for further discussions in the future. But in terms of outcome measures, we know that people are using many different outcome measures. Even for something really simple, something like baseline craving, we already have 24 different craving measures that people use among different trials that we have in addiction medicine. So that is problematic. So we are using many different measures, and there is no consensus about what would be the reasonable, even in a simple target like craving, just making a consensus about what would be the best outcome measure that we can make a consensus. So we realize that there is a kind of potential for an international network of tES, TMS trials for people to be able to do something together and face all those challenges that we have. And we had this experience before of having something like a textbook on neuroscience for addiction medicine. And over the time, we had 120 authors from 14 different countries in this kind of book that we recently published. And we realized that there are potentials for having people across the world contributing to these sort of activities. And we also had kind of some other experiences. For example, right now, the other thing that I do in my lab, we develop brain rehabilitation programs for drug users. And in this context, we develop kind of educational material and workbooks and the other things. And in the last International Society for Addiction Medicine meeting, we presented the material in Abu Dhabi, something like 10 months, 11 months ago. And over the time, the material were translated. Right now, we have them in 8 different languages. So we realize that people across the world, they are interested to adapt the material into different languages. So it seems that we might be able to do something together. But in terms of how we might be able to move forward for that, so what would be the future steps for moving forward for that? We are thinking how the next generation of studies could be replicable. So how we might be able to report the studies in a way that could be replicable. So how we might be able to have registration in a way that the studies would be completely automatically analyzed with pre-registered protocols. And how we might be able to use platforms like Open Science Framework together to share these pre-registered material. And it's not easy to do. I've done that, and it's really hard to define all these outcome measures, but it's something that we probably need to move forward. And there are already studies in Open Science Framework that they pre-registered the material and whatever they want to do to be able to reduce the chance of false positive or other kind of challenges in the field. And then we are thinking how we might be able to kind of promote online sync data collection platforms, how we might be able to have shared outcome measures together, how we might be able to even have shared protocols. And Flavio and Stacy are going to discuss about potentials for that. and then how we might be able to have a shared matrix of research, and how we might be able to also have multi-centric trials in the future. So these are potentials that we are hopeful to be able to do together. We had a meeting, a morning mixer meeting in New York City in August, kind of discussing with people, and we even had people from NIH there discussing about potentials, how NIH could contribute to the process. So we are discussing with folks from NIH as well, seeing how we might be able to have our standards as a part of their call for proposals in the future. And then we will have another meeting, we establish a neuroscience section for International Society for Addiction Medicine meeting, when we are going to have a meeting in Tucson. And also right now we have recently kind of submitted a symposium for the next brain stimulation meeting in Vancouver, and we are planning for a social meeting there for those who are doing addiction, trying to put people together. And we are going to have a special issue in Frontiers on brain and cognition for addiction medicine. But in this special issue, we are interested to receive publications in the field of addiction medicine. So that could be another opportunity. So it seems that there are potentials in this international network. And I hope that we can kind of move forward for these potentials in the future together. So that's the kind of the idea that we are right now trying to kind of promote. Okay, so we still have 2 minutes time before moving to the next step. Do we have any question before moving to the next step? Or any comment or idea?
Hamid, can you just clarify, you mentioned all the variables in protocol design. I think you also said that because of all these variables, literally no one has done the same thing. Is that correct? Like right. Once you add in drug, outcome measures, there's literally no consistency?
Right, and there is no replication. Yeah, that is something problematic. So if we want to move forward and being able to even make because there are right now even kind of 4 meta-analyses in the field, but the challenge is that you cannot put these studies together because the people are doing their own things in their own labs. So how we might be able to have shared protocols or shared outcome measures, that is one of the challenges that we need to think about together. Great. Flavio, do we have you online?
Yes, I'm here. Sorry, I'm struggling with my computer. Can you hear me?
Yes, we can hear you really well. So
great, because I'm on the phone, I'm using old technology.
Sounds great. Do you want to go ahead and you and Stacy discuss about what we have been discussing about following what Maro mentioned in terms of potentials for shared kind of platforms and also quality control standards?
Yes, absolutely. So Stacy had a loss in her family, and I don't think she is on this call. So I'll be trying to speak for both of us to the best I can. Welcome everyone. My name is Flavio. I'm at UNC, where I direct the Karana Center for Neurostimulation, and we're interested in mostly understanding how non-invasive brain stimulation modulates brain rhythms and how that translates to treatments for psychiatric illnesses. The way how I've been roped into this really amazing effort. So thank you, Hamid, by the way. This is really great. I'm honored to be here. Was through Stacy Daughters, who is professor of psychology here at UNC and has a long track record of studying. Hey, you're there. Hello.
I'm here. I'm just not on video. I'm here just for a little bit. But hi, everybody.
Oh, so then you can introduce yourself. That's even better. I was about to say lots of great things about you. Go ahead, Stacy.
No, I was enjoying this, so keep going. I want to hear what you said. No, go ahead, Fabio. Keep going.
All right. So Stacy and I have started to work together, and she's an expert and she can speak for herself, but essentially interested in changes in cognitive processes and how they relate to addiction and recovery from addiction. And my piece to this is the brain stimulation mostly focused on tES, so transcranial alternating current stimulation. We have some shared work that Stacy has been championing in that field. But I think today the idea is just to briefly talk about how we can essentially resolve the situation that Hamed has very nicely laid out. The field of non-invasive brain stimulation, in my viewpoint, carries incredible therapeutic potential because of its benefit profile, which is incredibly favorable. But I think we're reaching a point where initial excitement has switched to significant concerns from some parts of the research community, but also from funding agencies, to move forward and be able to help these patients. I think as a community, as Hamed has explained, we need to move forward in a way that we can provide answers which stand on really solid foundation. So I was thinking I would just take a few minutes to point out some things that we can probably consider as a community. Now, I think this is still early on, and typically early on in research fields, people have their own little idea, their own little pockets of money to try something. And I think that's how new ideas emerge. That's very natural, and I think it's unfair to criticize that. That's how this goes. If you have a new breakthrough idea, no one will give you money to do 500 patients in a first study. But I think there are two things how we can evolve together. One of them is that we share certain things across studies, which enables us to pull together data at the later stage after you have done your small individual pilot study. And the other one is following the direction that the National Institute of Mental Health has taken, which is really shifting from clinical behavioral outcomes towards neurophysiological and neuroimaging primary outcomes in studies. And that precision medicine framework, if you will, really enables us to learn a lot about how the intervention works and why it may have or may not have had a clinical effect. And so in other words, we're hedging against the negative results, which will become more prevalent, as we've learned in the presentation Hamed just gave, as we are increasing our end. So one thing which I would like to propose to the group is to think about what level of sophistication of brain measurements would the majority of us be able to do without significant extra costs or overhead, and to what extent could we streamline that such that even if many of the clinical aspects will probably remain heterogeneous for a while, we at least at the neurophysiology and brain network level have something to pull the data together. And I think there's going to be probably a conversation about EEG. There will be a conversation about fMRI, and there's also probably worth having a conversation about other physiological measures that some of you are interested, perhaps such as heart rate variability, other biological variables. And if we could agree on a set of and type of minimum standards that we try to fulfill in these measurements, and that we try to commit to using these measurements in our studies, I think would really gain some mechanistic basis for what we're doing, which ultimately will enable us to, through a rapid improve our stimulation paradigms. We are a very diverse group, which is great. So we probably have very different resources and expertise. I think one of the benefits of such a network is that we can share and kind of equalize that expertise. But my idea was mostly just have a conversation about, who is interested in EEG and where do we stand with that? Who is interested in MRI? Where do we stand with that? Who is interested in other physiological measures, and where do we stand with that, to see whether we could arrive at the minimum kind of shared consensus, how and what kind of biological data we want to collect in these studies. It's really more of a discussion than me giving you a presentation. So I'm curious to hear what the group thinks about that.
Flavio, can you talk more? I get what you're saying, that we're all going to do it our own way, right? Everyone has a little pocket of money, and you probably got that pocket of money because you said you're going to do things differently, and it's healthy. But is there, in your opinion, is there also a crisis that literally no one is doing the same thing?
Well, I think that's a great question, and probably people will have different answers to that. And it's kind of, I'm not prepared to make a value statement. I'm just taking the observation that early on everyone is trying their own thing, and based on what I've heard and what Hamed presented, I think maybe, this is a great question. We can make a step back and say, is it a time where we start to converge and drop our own individual little interest and start to do essentially, streamlined replication studies of the few main priorities? But in my experience it is really hard to get a diverse group of research to do that as a first step. So I guess I'm suggesting a more step-by-step procedure.
I just want to add, I think the issues that you are raising are important, and I think, given some time and exchange in this group, we will be able to, agree on some basic measures of measuring severity of addiction, for example, or using some kind of standard craving lists. Because we do have standardized lists for a lot of measures of interest.
Right. Yeah. So I think there's a lot of potential for streamlining also on the clinical behavioral assessment side. I just want to make sure we really try to think about the neurobiology of what we're doing, because ultimately that will be the make or break point for these kind of approaches, that we can demonstrate how they work. Because in contrast to pharmacological approaches, we're facing such a huge parameter space that we must understand the mechanism that we can optimize the treatments. And I think that's where we're different from pharmacological approaches, where we don't have all these choices. Thus, understanding the mechanism might actually be less burning. But that's my personal viewpoint. So
you're referring to the target engagement from the NIH, right?
Sorry, can you repeat that? Despite using old great technology, I didn't hear you. Sorry.
Are you referring to target engagement definition by the NIH?
Okay, I'm sorry. I think technology is failing me. Sorry. Did someone catch this? Can someone else repeat, please?
Target engagement.
Yeah, she's asking. You're referring to target engagement.
Yes, I'm talking about target engagement. Yes. So at the National Institute of Mental Health, you do not get the clinical trial funded anymore that has a clinical primary outcome. The primary outcome must be a biological variable, which essentially should demonstrate target engagement. And the rationale has been, we spent so much money on negative clinical trials without learning why they were negative and without learning anything about the underlying neurobiology. So there's been a dramatic shift, and our NMH fund, the clinical trials, have, for example, since we target brain oscillation, specific changes in EEG structure as primary outcomes. And I think that is a promising approach which will probably spread to other institutes of NIH. I hope I caught your question the right way. Sorry.
Yeah, absolutely. That's exactly what I was saying, and I think it's really important. I think that also, Hamed, with your PowerPoint presentation, which was great. Also, maybe it would be important to discuss proof of concept studies that mainly study target engagements compared to clinical trials with longer follow-up and more clinical outcomes.
Yeah, so I think there are many. There are many. Oh, sorry. Go on.
No, I just said, I think both are important.
Yeah, exactly.
Yeah, so I think there are a bunch of trade-offs here, right? So I think ideally all of us would like to see a study with hundreds of participants, which has done anything about the biological mechanism and the clinical outcomes, and even new treatment for patients. But we're very far away from that. So I think one way how such a group can be very effective if we even small steps, but we actually commit to them and we actually take these steps. So let's say we agree on 3 things that all of us are going to do in our studies, and when we publish our papers, we make reference to our consortium and say, here are those 3 important steps, and we've done them, and we report them in a standardized way. So then a year later, we have probably 12 new studies published as a group, and we can pull these 3 things together, and suddenly we're really hitting the non-linear synergy, if you will. So I'm advocating for small, solid steps that we can agree on to all insert into our studies well before we're starting to do long-range, big clinical trials.
This is Betty Jo Salmron at NIDA in Elliott Science Lab. We have been working on, we basically do get a standard set of imaging measures on everybody who comes through any of our studies. And the other thing we've been working on is developing a standard set of characterization instruments that we use, that are to be used across the all studies funded by NIDA. And so that might be something, jumping off point to talk about, common instruments that we might want to use across studies.
Excellent. Yes. Are you aware of something similar, NIDA, with regards to EEG?
Not now. We've been talking about it a bit, but yeah, we do EEG only in conjunction with MRI, and we're just getting up to speed on really cleaning up that data to be able to look at it. But we're getting more interested in using EEG as a primary marker also. So that's something we'd be interested in talking further about.
Right, I fully agree. I think one of the key aspects of non-invasive brain stimulation, or at least some forms of it, is that there's a very strong temporal structure which defines the dose in many ways, if you will. So understanding the temporal dynamics and how to change your stimulation would, and obviously I'm very biased if you know my background, I think that would be an important thing to also talk about, how to infuse that into studies and how to standardize it. Thanks.
And I know we're going to hear actually specifically about multi-center in a little bit, but I'm just wondering, just to throw out the idea, given our natural tendency to do things differently, are we only going to converge if someone like NIH or a company forces pays for it to converge? I mean, is it simply going to be that until either NIH says we're not going to fund anything unless you do it a certain way, or a company has to step in and say we're paying for it, so you're going to do it all the same? Are those things, are either one of those things necessary for us to practically converge?
Yeah, it's a great question, Merrim, and I'm not sure. I guess making a step back here is I'm not sure if it's time to fully converge, because if we're doing a large-scale multi-center study, let's assume funding available through a mechanism you outline, would we as a group know which out of these different many things we would want to pick? And if we don't, then I think we're probably not ready for that, because we don't yet understand enough how this actually works.
Well, I would add too, just that the field of addiction generally hasn't converged on kind of the ideal outcome endpoint. Forever it was just abstinence only, but now there's increased attention to reduce drinking and reductions in cocaine use, reductions in other endpoints. So I think this isn't unique to adding neuromodulation. I think this is an issue generally in the addiction field. Certain aspects of treatment would target cognitive functioning, other aspects would target craving. So I think it really depends on both the substance and the goal of the patients, as well as what you're trying to change. So I see this as, again, I mean, NIDA is working towards making some consistency across trials, but generally that this is a problem in the field of addiction broadly.
That's very helpful. Thanks. That's a part of the field I'm not very familiar with. It's helpful to know. Thank you.
Yeah, no, Katie, that was really well said. It's true. Addiction, there isn't convergence on what the important outcomes are, what the mechanisms are. So this isn't neurostim specific. It's addiction, an issue in addiction broadly.
And thanks, Stacey. And I'll add to that, addiction's it's a complex disorder, and so it's multidimensional. People come to it from different pathways, and so I think there is complexity in the disorder too to consider.
Right. I mean, I think we've heard kind of both perspectives. Is a really helpful discussion. One is this is really complex and very heterogeneous. There's no way we can converge. The other one is to really get any solid, convincing data. We need a large study, which necessarily needs some pronounced amount of convergence. And I think both perspectives are incredibly valid and helpful, though probably not resolvable. So. So I think that goes back to the idea: can we just start rolling out first small steps, which do make sense across this very heterogeneous disorder, that makes sense for all of us to collect in a standardized way and report in a standardized way to to achieve a first step of convergence, and what could that be? And I know I'm kind of going back to the same point 3 times, but I think my desire is that at the end of this call and in general through this, it's just not one more thing we're doing, but really there's something concrete which takes a little time, but it really makes everyone's science in a synergistic way better. And I think a few small concrete steps would be, from my perspective, a good middle road to go for now.
Any other comment from other colleagues? Good. The other thing that we are already discussing with Flavio and with other colleagues like kind of Felipe Fregni, and I think that Felipe is not with us today. He was supposed to join us for the meeting, but I cannot see his name right now among participants. So we are discussing about other than outcome measures that we are thinking together, and I'm going to share a document with you in coming days in terms of the outcome measures that people have used till now. So what people have tried to explore right now, and then we can start to discuss what we already have and think about what could be the next steps of merging some of these measures together and make a consensus together. But in terms of the next steps, we are also discussing about a checklist of quality measures for trials, how people are going to design the trial and how they are going to publish the trial. And we know that there are already some checklists available outside, like the charge checklist by people from Cochrane in terms of the quality of kind of reporting a trial. But there are some minor details that are related to non-invasive brain stimulation or are related to tDCS or TMS in specific, or are related to addiction medicine. So we are trying to add those items to the checklist. And we have already started to kind of circulate a list of items. And Flavio suggested a checklist that they developed before. So we are discussing about kind of starting to share a checklist of quality control items that people might be interested to consider when they are publishing a trial in the field or designing a trial in the field. And we are right now thinking about kind of receiving feedbacks from people who are in the call right now to see what are the quality measures that they like to keep in the list or they like to kind of remove from the list. So we try to make a consensus. It's not going to be easy, but we try to see how we might be able to move forward for these two major kind of target in the network. One would be how we might be able to have shared protocol and shared outcome measures, and the second would be what would be the quality and standards for designing and reporting a trial in a way that could be reproducible in the future. So these are two targets that we are hopeful to be able to do together in coming weeks. And in terms of the next steps for being on time, we are also going to kind of think about safety measures. And Shirley and Marom are going to discuss about kind of safety measures. And I know that both of them have done lots of different things for safety measures. So Shirley, Marom, do you want to kind of start and kind of introduce the idea of how we might be able to be in the safe zone for addiction medicine in the future?
Yeah, I can start. So as we discussed also in New York, first, of course, we need to describe the side effects of non-invasive brain stimulation in general, you know, that it's addiction or any kind of population. And then we also discuss about discussing safety per types of addiction. So it's going to be very different from tobacco to alcohol. Also in regards to stages, and those who are experts in addiction, you know, you can help us here, but also in terms of stages, are they abstinence, are they still using the substance, are they abusing the substance, and age. As well as we know in addiction, it would be good if we can target younger individuals before it's too late, where really the brain is impacted by substance abuse. But of course, using our TMS or tDCS in children, it's not clear yet in terms of safety. So I think we should discuss that as well. And then we discuss safety monitoring, as it's usually done for these patients from medical doctors in addiction, and also monitoring of substance use and abuse during the non-invasive brain stimulation regimen. So having some control of, you know, where they're drinking between RTMS sessions and stuff like that. So these were the main topics we discussed in New York that we thought about. I don't know if Maran, you want to add some things?
You know, I think there's been work on tDCS safety, and I guess the bottom line is because there is no known serious adverse event, we don't know where to draw the line, you know, and with TMS, people just, they know seizures, and so that's what they focus on, right? And as long as you can avoid seizures, right, that's the assumption, you're more or less okay. And with tDCS, we don't have that. And so it leads to a lot of ambiguity, and you know, but anyways, there's major efforts on safety for TMS and tDCS already. And so I guess for our purpose here, we just need to color those. Are there any special considerations for the populations we're dealing with? But other than that, I guess we don't need to reinvent the wheel. We can fall back on the standards that are in place.
Yeah. So just speaking to that briefly, I mean, one concern we had in our trial with alcohol, people who were actively drinking, was alcohol withdrawal and potential seizures in alcohol withdrawal. So we excluded any individuals who had any symptoms of alcohol withdrawal and did a withdrawal assessment before every tDCS session. And I think that's it. Thinking about the withdrawal syndromes for these disorders is important.
Yeah.
Any other comment from other colleagues regarding the safety issues? Any experience, any concern, any question?
So we've been running TMS studies in cocaine, in active cocaine addicts. Started with inpatient to try to get a better control over their use, but we haven't required them to be urine negative. So we are administering ITVS to cocaine addicts who are urine positive still, not showing signs of acute intoxication, but urine positive. And we've not run into really any significant problems with it.
I can confirm here in Italy for our patients that are cocaine addicts, and basically over a follow-up of more than 2 years, we are not experiencing any severe side effects, even with the we have just reported from a slight headache, but no other side effects, and are also temporary. I mean, also for example, the headache doesn't last longer than a couple of days. So this is our experience.
I'm working on the tDCS, and I found there are some effect. The effect size of the tDCS should be less than the smaller TMS. But we use the 2 MA, 30 minutes. But I look on paper. Some papers show that we can use the 4 MA. But if we use the 2 MA, some participants report they feel very not good. For example, the flash and some pain. So I'm not sure which the level, which the stimulation should be used. Which level, the 2 MA, 1 MA, or the 3 or 4 MA? I'm not sure that. I think this is very important for the tDCS issue. Is that?
So this is Maroon Bixon. Can you hear me? Sorry, I had to switch to the phone. But hello?
Yeah.
Yes, we can hear you.
Okay. So yes, there's been work with tDCS obviously to go up to 4 milliamp, and there's also been work obviously on new TMS protocols. And I think just one way to approach it is to say we have doses that we are comfortable with making consensus statements about safety, right? Things that have been FDA, you know, in case of our TMS, things that have been FDA cleared. In the case of tDCS, talking about 2 milliamp. And when you go to new doses, like frequencies and RTMS that have not been commonly used, or 4 milliamp tDCS, we just don't have the data. And so you sort of have on the one, you draw a line, and on the one side of the line, you have things that you feel there's enough data to make a statement about safety. And on the other side of the line, we just don't. And it's fine. That's sort of in this investigational category. And that just means that, you know, every trial needs to do a risk assessment, which you do anyways with your IRB. So that's maybe one way to just kind of just recognize we have things we can say what's safe, and there's just things that we just don't have the data. And that doesn't mean they're dangerous. They could even be safer, but we're not going to go there because we don't have the data.
So if I think about that, with the trials that we have right now, we do assess side effects related to brain stimulation for at each session, but we also have an additional side effect form related to the withdrawal symptoms, to the symptoms really for this, right now it's tobacco addiction. So, so we add a second side effect form. So I think in the future, we'll have a better idea what is really related to tDCS and maybe more related to a specific addiction.
Hi, this is Greg Salem. My video is also not working as well. You know, I think obviously this is a really savvy group with all this stuff, but just I'm not sure how many people clinically use TMS. And I can say in depression, aside from being worried about seizures, certainly we've seen some people clinically with a manic switch if they're bipolar, and we've certainly seen some people with psychosis get worse. And so, you know, when designing trials, it's just probably a good thing to keep in mind for those of us who don't do a lot of clinical work with TMS. So I just want to throw that out there as a couple of other concerns that folks may want to consider.
We have seen someone get hypomanic with just a fairly brief exposure to the H coil, so that, and it was not someone without any risk factors for it. So it is definitely something to keep an eye out for.
Great. We still have 1 minute left for the safety issues. Do you have any question or kind of experience to report on safety issues?
Exactly. Probably another aspect we should think about is about the session of stimulation and the number of sessions and the side effects we have. Because most likely we still don't have this information. So, for example, there are patients who have been treated with 2 sessions per day for more days consecutively, and they can report some side effects. And other patients following probably a treatment with a few number of sessions. And so probably this is also another aspect we should think about in this network and gather all our experience.
Great. So it was a really nice discussion. But just to move forward for the next step, the next presenter would be Dr. Zangen from Israel. So Abram, Bumi, how are you doing?
Hi, thank you. I'm fine. I'm sorry I came in a little bit late meeting, so I was hearing Flavio talking, and I'll have to comment on this. I was, so I didn't want to interrupt, but I have a different view on some issues. So I'll have to start with that before I get into the multi-center suggestions that I have from my experience.
Sure. We have 10 minutes for you. Just go ahead, and we are all ears.
Yeah. Okay. So, well, my view is that we have, if we really look at the literature, and I think I was one of the first people to really start that in an animal study already in 2007, published in Journal of Neuroscience, looking at animal models and looking for how brain stimulation can affect plasticity markers in addiction and how this can change behavior. And since this, I think, first paper in the field of brain stimulation in addiction in 2007, and this one in Journal of Neuroscience, there were so many other studies, both in animals, then came later on the Antonello Bonci's paper, optogenetics, and other mechanistic studies. And I agree, we don't have a full understanding of mechanisms, but I think that in medicine, you have so many examples of cases where, just take ECT as an example, so many examples of cases that you don't really know the mechanism until today, like with ECT, which is used for almost 100 years and is the most effective for depression. And still, there is a place for multi-center studies, for research, to different parameters, different ways to do it. And it's a very effective approach. And therefore, I do think that it is necessary to try and find some coherent way of doing the measures and using specific brain stimulation parameters based on the literature view, and then to advance the field. Because just saying, we first have to really understand the mechanism and not moving forward with trying to help patients, I think will get us to a place where we can stay with the basic questions for another 100 years and not really advance, not really even get, I mean, from a moral view, we won't even have the ability to request funds from governments to really continue this. So we do have to do these things in parallel. We have to understand the mechanisms better, but we also have to do studies. So I disagree with the messages from Flavio earlier. I heard it only want to interrupt, but that's just wanted to make this comment very clear. So again, starting from animal, which back in 10 years ago when we really all started it, which is very simple. You can do a very easy multi-center study with animals because you have very clear methods and you have very concise, I mean, ways of measures, objective measures of animal behavior, and you have a very coherent population. Then, since then, I was involved in 6 different studies in clinical studies in humans with TMS in addiction. 2 of them I was a PI in the smoking cessation studies published. And I think that the main take-home message we learned, and I guess you talked about, is the need to have objective measures, because the subjective measures of craving, many other different ways of measuring the subjective experience of addiction are all more variable and there is a big gap in our experience, for example, between what people report about their smoking behavior and their actual cotinine levels. We find effects based on the subjective reports, but when you measure cotinine in their urine, you see that they continue to smoke. They say they quit, or they say they reduce their smoking by so much, and you can look at the published papers we have. And then when you measure the cotinine levels in their urine, you see that they smoke more. So we did some more research into this actually, and got back to some of the patients, and then we understand that in some cases, in the case of cigarettes at least, they probably do smoke less cigarettes as they report, but they inhale more, or they smoke them all the way to the end. They smoke more, and therefore you don't see the reduction in the cotinine, but they do report less cigarettes being smoked. But after all, you know, the ultimate goal, as we all know, is to have a complete quitting of any drug abuse, and if you just reduce the abuse, come back to the original level. So I think this is a very critical aspect that have to be measured in multi-center studies. This would be the primary outcome, is complete quitting. It will be very interesting as exploratory measures, as we have now in our multi-center study in heavy smokers. It involves 22 centers in the world. This is following our study published in Biological Psychiatry using the H coil, the H4 coil for targeting the insula and the ventrolateral prefrontal cortex bilaterally for smoking. And there we learned that the cotinine level it measures are great, and all these secondary measures like craving are great to have, and I think eventually the goal, as at least FDA would require, would be complete quitting, and this is the measure that should be the primary outcome measures in any multi-center study, is complete quitting and measured over time. So in the case of multi-center study, the primary outcome is quitting for at least 4 consecutive weeks. This was the requirement by the FDA, so at least in 4 consecutive weeks, the subjects report that they quit the smoking, and also confirmed by creatinine levels in their urine. So you need to take frequently enough samples based on the half-life of the metabolite that you are measuring, so to really confirm a consecutive period of 0 amount of drug in urine or blood or whatever sample you can get for that. So this is my suggestion about multi-center studies. You have the primary outcome being quitting and having a longitudinal measure for that. The other issue, of course, is that the other challenge we aim when we have 22 different centers and now trying to confirm a study that we published originally in a study in one center in Israel in smokers, is to have a similar type of populations in different centers. And I think there is always a component of the psychological support from the team. So you have this operator daily coming to stimulate the subject, and there is an interaction, 50 minutes every day, and you need the operator to be blinded, and of course the people who rate all the, at least the interviews the raters should be blinded to the treatment arm, whether it's an active or sham stimulation. But I think this interaction between the operator and the subject daily should be, on one way, should be blinded, of course, but should always be supported, because we need this psychological aspect in the treatment. And basically, if I can add here my view on the field altogether, is that our stimulation can only open a window for change, for plasticity. And this is also what we saw in the animal work, that you do make, and today we have some electrophysiological measures suggesting really long-term changes in physiological measures. I cannot get into the details right now, but this only opens a window for more delicate change in the circuitry. And the delicate change in the circuitry would be a result of this interaction between the subject and the operator daily. So therefore, I think this the way to make in a multi-center a very coherent approach where everyone receives the same attitude, then I think for the next study, we don't have it right now in our multi-center study. It would be great to have some application in a smartphone, or at least instructions, written instructions, both for the operator and for the subject to follow during the period of the stimulation. So I think my time is up. I just spoke for my 10 minutes. And so my main take-home messages is the outcome measures being quitting for a longitudinal way for a long time, and trying to have the subjects experiencing the same in all centers is very challenging. It requires training. And of course, when we do this multi-center study, which is FDA regulated and funded by a company, then we have this investigators meeting, and we can, of course, go over all the details of the protocol and put emphasis on the approach of how to really approach the subjects during simulation, what exactly to do, what is the timeline, and have as much as possible conference calls that we do have during the multi-center study among the PIs, and trying to be in touch with the operators in order to receive feedback along the 2-3 years of the study, and try to homogeneous as much as it is possible in the 22 centers that we are doing right now.
Thank
Any
you.
questions? I'll be happy to answer.
Thank you very much for being on time. We can have one question from Abraham. Do we have any, just one single question? Any question?
A quick question. I think abstinence in a substance like nicotine, which is directly harmful in lower, smoking, which is directly harmful in lower quantities, is appropriate. But I don't think abstinence is necessarily a required outcome in something like alcohol and cocaine, other issues, other substances. But another thing that we've run into in our smoking cessation studies with using the Nic Alert, and as you mentioned, Bhumi, sometimes people report they're not smoking, and you get your Nic Alert back, and it's still a 6. And we've had the luxury of being able to send these out for confirmatory mass spec assessment of the nicotine cotinine levels, and we've got a number of examples now of people dropping a 6 for weeks, and the confirmatory mass spec one says that they're not smoking. So I'm not sure what's going on with that. I haven't been able to fully follow that up, but we've had issues with the Nic Alert not giving accurate results.
So you look at your cutoffs, because sometimes the
we looked at the cutoffs, and we compared the cutoffs that they claim to be using for the Nic Alert, and when we get the concentrations back from the confirmatory tests, they should be dropping 2s and 3s.
This is Flavio. I just have a quick comment because I was honored by receiving feedback on what I said by you. I just want to make one quick clarification. We might disagree on how to proceed, but the statement that I'm not interested in helping patients or want to delay helping patients is a very inaccurate and wrong representation of what I tried to say, and I'm sorry it didn't come clear to you.
Okay, I'm sorry I understood that this way. I said I came in the middle of your talk, but also that's why I didn't. It was not personal, Flavio.
Well, I think that's a very good point. We need to be gathering enough data from these clinical trials to be able to enhance our understanding of the disease and what things may or may not work at different times.
If I may make a quick comment, Ahmed. Thanks, Abraham. It was great, and I, fully agree with what you said about objective measures and quitting and reduction. But quitting is better. I do think that craving is also, even though it's self-reported, it's subjective. I think it's still important to measure, at least as a secondary or third outcome, because it's one of the main factors of relapse, and we cannot ignore relapse in addiction. So, I'll just make that comment. Thanks.
Great.
And this is Maron Bixton. I just wanted, I know we were running over, but I really, that point that Abraham brought about neuromodulation opening the door, I really liked it. This is a theme that in tDCS is becoming severe, that the patient experience, everything else other than the neuromodulation, has a huge effect both on the size of the control arm and on what the neuromodulation itself does. And if that is not controlled and documented, things may actually not be reproducible. So I think that it's an easy thing to do, and I think I think it's a great control the patient experience from the moment they walk into the door, where they sit, who talks to them, what else, the whole environment could be critical for reproducibility.
Yes.
Right.
And particularly in a time where placebo effects are huge, and where once people decide that they want to or quit, they're going to be doing that not modulation and treatment, but on their own. And so that's really important to keep in mind, that even their interactions in many large trials, patients actually remember the assessors more than the therapists. So it's important to keep in mind that the person interacting with the patient is going to have an effect.
And one thing also that we discussed 2 years ago at the workshop on tDCS at the NIH with Sarah Lizambi. So we talk about level of expectancy. How much a patient thinks that he's going to succeed by of quitting. So I think that's also something that at least we are measuring. So we ask, from the beginning of the IBS regimen, how do you think you're going to succeed? Do you think you're going to be able to quit? And what's the level of motivation? So I think this is also important.
You will not have, usually you will not have subjects clinical study which requires multi I mean, every day, coming for many days, for a long time, if they're not highly motivated. So originally, if you don't have high dropout rates, you will need to get these highly motivated subjects to begin with. And
Yeah.
you will.
I think it would help us to explain the attrition rate, you know?
Yeah.
Okay, great. So let's move forward. And I think that sometimes these kind of informal discussions would even be more kind of helpful for further collaboration. And I think that we need to keep on discussing about these things that are super important. So just in terms of the timing, we would move forward and ask a few colleagues to introduce what they are doing in their own labs and kind of discuss about potential for future collaboration. The first one would be Esther from Brazil. Esther, do you want to move forward? I'm going to share the slides that you kind of sent me. So I will share your slides on my screen, just getting sure that you will have the slides as well.
All right, thank you very much. And it's very nice to meet you all. And it's been very exciting to have this discussion. I'm Esther, I'm from Vitória, Espírito Santo, from Federal University of Espírito Santo here in Brazil. And we have been working with mostly with tDCS in alcohol use disorder and also crack cocaine use disorder. These are both very difficult conditions, as you all know. And I was worrying to talk about quitting, completely quitting, and I'll just pick in this because I'm learning that drug dependence, they are similar in general, but they have specificities, peculiarities, I mean, from each of the drug dependence. I'm learning now that we cannot have the same result with alcohol use disorder and also and crack cocaine disorder as we are getting the new results now. And if you can go through the next slide, please. I won't be showing, I won't be saying all the details, but these results shows the relapses and craving results. We picked this as primary outcomes in our studies, but we do also have electrophysiological results and other cognitive measures, other clinical measures, quality of life also. Okay, this is just the first study we have with alcoholics in outpatient service, and we measure relapses, and we found after the 6-month follow-up that we had smaller relapses in the patients that received tDCS, real tDCS. And in the bottom, we have a craving measure in the crack cocaine users. And also, in this study, we didn't have a follow-up, but we do have in the next study. If you can move to the next slide. We run the bilateral stimulation over the dorsolateral prefrontal cortex, right? And we are placing the anodal to the right and the cathodal to the left dorsolateral prefrontal cortex. And these are recent results, and we just published the result from our alcohol use disorder on the top. And we saw differences on craving measures by the end of the 10 sessions. In this study, we use 10 sessions. And the interesting thing is that the relapses, they relapsed less after 60 days of follow-up. But the crack cocaine user, they didn't. They didn't differ from the shun group, and they relapsed a lot. I mean, in a similar way from the shun group. So this is all our recent results. We didn't analyze it, our electrophysiological study yet, okay?
Great, thank you very much, Esther, for presenting what you are doing. A great job that you are doing in Brazil. The next speaker would be Roth Van Holst from Netherlands. So, Roth, would you just go ahead, present?
Yeah, thank you. Yeah, so my name is Ruth Van Holst, and I also represent Anneke Guderian, who had to just leave us. But I want to introduce you to our small research group focusing on RTMS mainly. We're based at the Academic Medical Center at the University of Amsterdam in the Netherlands. And our small group contains of Anneke Guderian. She's Professor of Working Mechanisms and Addiction. And I am a PI, and I focus on investigating with fMRI and PET the neuromechanisms of addiction, and also use RTMS to do so. And we both, so I think the slides are gone. I don't know if that's a problem. Can you see the slides still?
Just one, okay.
Yeah, thank you. Okay, so currently we have one PhD student who is doing a quite large RTMS clinical trial, in which we investigate alcohol-dependent patients who receive cognitive therapy, and add on, we give them 10 days of RTMS simulation. And so we have 40 people who receive placebo and 40 RTMS on the dorsolateral prefrontal cortex. And a part of them we also scan with fMRI to look at processes such as impulsivity, brain connectivity, and our main outcome is actually relapse rate. Because we do think that craving is very important, but we would like to see how it actually enhances people's treatment outcome. So this is what we're doing at this point. The details of this study was published in the BMC Psychiatry. René Schluter, so the PhD student who's running the study, published a review on the effects of neuromodulation on cognitive functions in addiction. And I think that I was actually surprised that we focus a lot on craving, but it's hardly known, as was also addressed, how this actually would work on neurocognitive basis, really. This is what we're doing, and I hope this was a short introduction.
Thank you very much. The next speaker would be Dr. Jonathan from India. Jonathan, would you move forward?
Hi. Yeah, so I am a faculty at the National Institute of Mental Health and Neurosciences in Bangalore, in India. And I'm primarily working as a clinician in the context of obsessive compulsive disorder. I'm affiliated primarily to OCD clinic at NIMHANS, as well as to the neuromodulation program at this institute. I also represent our team on behalf of Dr. Venkat, who was supposed to be present. He's on travel now, and Dr. Shiv Kumar, who's present in this panel. So our lab has been conducting studies for the past 5 years, mostly in the disorders of schizophrenia and obsessive compulsive disorder primarily. And there are some studies, I should admit that the studies are very few in number in the context of addiction, and we would like to take it forward and conduct more studies in the context of addiction. So we've been using tDCS as a primary modality for the neuromodulation aspects. And we have done, as far as the safety aspects, which we were discussing some time back, we've been doing tDCS for quite some time, multiple sessions without major concerns or any adverse events that required termination of the tDCS session so far. And can I move to the next slide, please? these are some of the studies which we published recently in schizophrenia, and some of the randomized control trials. One randomized control trial which we completed in the context of obsessive compulsive disorder. And in the context of addiction medicine, the studies has been very nascent. I would say it's been just in the preliminary phases of the subject recruitment. In the next slide, yeah. Thank you. Yeah, this is just an initial study wherein we, in addition to the clinical outcome, we try to examine what are the resting state measures that might predict the outcome in a very small sample at this point of time. So we're looking at continuing this particular study. This is a very preliminary study which was done. So I'm looking forward to this collaboration. Thank you.
Great, thank you very much. Okay, the next speaker would be Dr. Katie Witkiewicz from University of New Mexico. Katie, would you move forward?
Yeah, yep. Hi, everyone. So yeah, I'm at the University of New Mexico, and my background is mostly in treatment of addiction from using behavioral interventions. And so given a collaboration with Vince Clark and Eric Claus, who might be on the call as well, we started working with seeing if we could add tDCS to this intervention. So next slide, please. So just a brief bit of information about mindfulness-based interventions. So we've been using mindfulness for almost 18 years now to treat addiction. We combine mindfulness with skills training groups. This is done in groups and led by mindfulness practitioners who are also trained in treating addiction. It's modestly efficacious for addiction, effect sizes in the small to medium, and better than cognitive behavioral treatment. So I think that's important. It's a better intervention than kind of gold standard care. However, treatment engagement and completion is somewhat lower. So we thought about adding tDCS as a way of enhancing engagement in the mindfulness treatment. Next slide, please. So the basic idea with this was Vince Clark's prior studies that targeted right inferior frontal gyrus with 2 milliamps of anodal stimulation. Basically, people reported greater mindful awareness. And certainly when I tried it for the first time, I was pretty impressed with the ability of tDCS to kind of enhance awareness of present moment experience. And for those reasons, we moved ahead with targeting right inferior frontal gyrus with anodal stimulation. Cathode was placed on the left upper arm. So next slide, please. So we've run 2 trials now. The first study was with smokers, all active tDCS. This was an open, not blinded trial with 8 individuals, and we found large effect size reductions in cigarettes per smoking day, and actually a 33% biochemically confirmed cessation rate. They all relapsed to smoking within a short follow-up, and so we looked at their drinking, and their drinking actually decreased. So that led us to apply for R21 funding, which we just actually recently completed this month on the right there. So this was a double-blind trial of active 2 milliamp tDCS, 84 individuals. Importantly, these are people who are actively drinking and came into the trial to reduce their drinking. So similar to what Betty Jo said, this was a harm reduction trial. It was not based on people trying to obtain abstinence. It was based on people trying to reduce their drinking. And what we see here, I mean, these are very preliminary results. I just got the data entered last week. But during treatment, we see about a drink per drinking day reduction in their drinking that was maintained better over a 4-month follow-up in active tDCS than sham. So, we're going to be putting together these results soon and analyzing them using the pre-registered clinicaltrials.gov analysis plan and there. Thanks.
Thank you very much. The next speaker would be Sarah Hermans from Belgium. Sarah, are you there?
Yes. Hello everybody. So my name is Sarah Hermans. I'm a psychiatrist in the University Hospital of Ghent in Belgium, where I'm a member of the lab there, where the principal investigator is Chris Baken, but he couldn't make it. for today. And my main interest is in TMS and alcohol addiction. We had some previous, so the main interest, the main focus of the lab is depression, but there are some projects regarding addiction. And the previous projects were that we tested, we evaluated a single sham-controlled TMS session in alcohol dependent patients, and we assessed their craving in their home environment, and also we assessed their cognitive control. Then we performed a study where accelerated TMS was tested, where we administered 15 sessions during 1 week. And we assessed brain activation during acute exposure, also voxel-based morphometry and craving and relapse. The last study that is almost finished is the sham-controlled bifrontal tDCS session in binge drinking students, where we also evaluate craving and cognitive control. Can you go to the next slide? Hamid, can you go to the next slide? Thank you. And what is coming up, which is starting up, is the evaluation of a single sham-controlled tDCS session in acutely hospitalized alcohol-dependent patients. And since we have also a collaboration with the lab in Brussels, there will also be a study in cocaine-dependent patients, but it's not sure yet what will be exactly performed. And that's about it.
Thank you very much, Sarah. The next speaker would be Dr. Martin Hermann from Germany. Martin, are you there?
Yeah, I'm here. Do you understand me?
Yes.
So you can switch to the second slide.
Okay.
Oh. Okay, we did 2 studies in addiction. The first study published by Bicharke was in alcohol-dependent inpatients, and we did only 1 session tDCS, and we measured psychophysiological responses like the startle response, and we also used subjective craving rating, and we found this 1 session tDCS changed the processing of emotional cues as well as subjective ratings. The second study published 2 years earlier by Alisa Dila was in smoking people, and we added, or we combined psychotherapy with TMS stimulation, and we found a change in abstinence rate after 3 months. So this is our 2 studies in addiction. Slide, please. The next slide, please. Thank you. So we focused on anxiety disorders the last years, and here we try to focus on processes which are relevant for anxiety disorders, like fear extinction processes. And we found that we can stimulate medial prefrontal cortex with rTMS, and we improved fear extinction in the lab. And then in the next study, we also showed in a controlled trial that this rTMS in combination with exposure therapy improves therapy responses. And we want to continue in tDCS studies and showing the same. So we try to find some electrode placement for tDCS to augment fear extinction in tDCS. And this worked. And I think for me, or the work in anxiety-related things is that we try to focus on processes. And this might be interesting for alcohol-dependent patients or dependency addiction too. So to combine it with psychotherapy or some therapeutic approaches. That's my background. Thank you.
Thank you, Martin. And then we are going to have people from Elliot Stein's lab. I'm not sure who is going to present from Elliot Stein's lab right now. Who is
yeah,
on?
We're all on Betty Jo's feed, so.
Okay.
Can you hear me?
Let me just bring up the. Okay.
Yeah, so I should start that we're all from NIDA and Elliott Stein's lab. We'll have 3 studies we'll try to rip through quickly that we're doing with neuromodulation. And also I spoke with Colleen Hanlon today, and she wishes she could be here to be part of this conversation. She'll be in other conversations.
Yep.
And so, yeah, so we'll start with my study here, which uses an accelerated theta burst stimulation as a clinical trial to try to reduce cocaine use. Again, we're doing harm reduction, but we could measure any kind of abstinence as well over a 6-month period. So we're collecting healthy controls and cocaine use. We get full characterizations of clinical and neuroimaging measures before we start any neuromodulation. And you can see that in the week 1 first couple visits. We have a couple scans and the clinical measures before anything starts. And then we start 2 weeks of ITVS treatment, where we do 3 ITVS treatments a day with 60 minutes between each treatment in accelerated fashion to get to a magic number of 30 sessions over the 2 weeks. On the first day, we would measure MRI before and after to get acute stimulation changes within cocaine users. Healthy controls would only do 1 day in the crossover design, active and placebo theta burst, whereas the cocaine users are split into separate groups and they get the full 2 weeks of treatment. All cocaine users get the contingency management that you see there for the 12-week period as a treatment as usual. And then we do several follow-up MRI scans to assess any kind of neuroplastic change, in addition to the acute neuroplastic change we would get on day 1. And we do many MRI scans where we're targeting all the circuits that we're interested in, the craving, reward processes, executive control, and we can get at, as well as other more structural DTI, arterial spin labeling, and the like. So we're trying to do a full characterization of these participants as they go through treatment and how it relates to drug use behavior, and then also urine testing and the like. So that is a quick overview of my study. And I think we're going to musical chairs, and Sarah will do the next slide. She'll talk about her stuff.
Hi, everyone. My name is Sarah Aronson Fischel, and I'm an MD PhD student here, so doing medical school at the University of Maryland. If you could bring up that slide that you had before. Yep, that one. So this is my thesis project studying the effect of transcranial direct current stimulation simultaneously in the MRI scanner. And I'm specifically looking at the nicotine withdrawal syndrome, which is the most common time period in which smokers fail their quit attempts. And so my study is based on the tripartite network model that our lab and others have developed in its application to drug addiction and specifically nicotine addiction. So I'm targeting 2 nodes, one of the default mode network and one of the executive control network. And to do that, I have 3 different montages for tDCS. So one has the cathode over the ventromedial prefrontal cortex, which is a node of the default mode network. So the idea is to dampen down the default mode network, and the other is the anode over the dorsolateral prefrontal cortex, and the idea is to bring up the executive control network. And then we compare that to where the polarity is switched between the 2 nodes, and then also there's a sham condition. And we also test this with smokers and non-smokers. For smokers, we have an abstinence condition where they wear either a placebo patch or a nicotine patch. And when they, all these participants come in, they do tDCS simultaneously in the scanner, and we have scans before, during, and after stimulation. That's a really quick overview of my study.
And one more. I'm John Pedota. I'm a staff scientist in the lab. In addition to the studies that we're doing, like Vaughn talked about, looking at treatment, we also want to take a crack at understanding the mechanism of TMS and relevant mechanism for acute administration. So on the left, we've identified a circuit independent of TMS that's modulated as a function of abstinence. And so the anti-correlation between the ventral insula and left dorsolateral prefrontal cortex correlates with subjective craving during abstinence. So the more anti-correlated the circuit is, the more craving the individuals report. The second figure there on the left shows the location of the circuit that we've identified in relation to the magic F3 spot for TMS. And what was striking to us is the spatial similarity between the two. And as I'm sure many of you in the group know, there is a lot of evidence suggesting that left DLPFC TMS assuages craving in smokers. And so basically what we're trying to do is create some causation between the correlations that we've seen. So the idea is that if we take smokers and scan and give them TMS both during satiety and abstinence, and then put them directly in the MRI scanner after that, we can see if the TMS that we anticipate will alleviate some of their craving also modulates the circuit that we know is related to craving, and then that would give us some level of understanding of why we're seeing the clinical effects that we're seeing. And the overall idea is that the state that you give someone TMS in matters, right? So in this case, potentially abstinent smokers, those with this circuit dysregulated, may be more likely to respond to TMS than non-abstinent smokers are. And we'd have now an objective readout instead of a subjective readout in the strength of this circuit. And in addition, we'll give them a number of other measures of things like cognitive control and just general resting state, functional connectivity, to see other downstream effects of a single acute administration of TBS in different states of nicotine presence or absence.
Great, thank you. Thank you very much. Okay, now we have time for others. Right now, I can see that Michael Nietzsche is also with us. So, hello, Michael.
Hello, sorry for being late. I had another for European project, so couldn't come in time.
Oh, thank you very much for joining. Do you want to do any kind of comment on what we are discussing and based on what we have been discussing before?
I was just joining some 15 minutes ago, so saw the last presentations, which showed nice, interesting, ongoing trials. But can't comment about the things which happened before. Sorry.
Great. So let's just see how we can change this one. Okay. So now we have time, kind of few minutes to see if any other person in the meeting would like to introduce what they are doing or kind of discuss about questions that they have, and then we will have a conclusion and wrapping the meeting.
Hi everyone, maybe I can introduce myself. I am Samir from India. We had done some work using TMS in alcohol, mainly for craving. We have 2 published studies in that, but currently I am working mainly with tDCS, not with TMS. So we are working in field of there's other disorders like schizophrenia. We have worked on somatic symptom disorder, OCD, and I have an upcoming project on alcohol. So that is the reason maybe I'm here. So we are going to have a project on alcohol using tDCS. So I have identified 2 specific positions which would be useful for craving as well as reducing risk taking. So, and I'll be running some markers as well, possibly some blood markers as well as electrophysiological markers. So it is still in that preliminary stage, so yet to start the project. Thank you. And I think, yeah, getting a consensus on the various aspects of this would be really interesting. I will look forward to that. Thank you.
Great, thank you, Samir, for joining us. Any other person who wants to present or talk or?
Yes, I would like to introduce better our clinic, our foundation. We are based in Italy, in Padua, and I'm speaking on behalf of Dr. Galimberti and also Dr. Bonci, because we have this active collaboration at NIDA. I'm a neurologist, and now I'm scientific coordinator here at Novella Fronda Foundation, and basically we have three main areas where we are working on applying rTMS. So we are working with cocaine addicts in alcohol abuse disorder and gambling. And we are focusing, of course, on the effects of our TMS on outcome measures we are discussed during this meeting. But also we are investigating also the role of other areas in having an effect on these disorders. And we have also a pilot study ongoing, focusing on the SMA, applying a low frequency stimulation protocol. And also we are using other techniques to possibly target and address these neurophysiological changes by using exactly neuroimaging and also NIRS. So this is our main work.
Thank you, Graziella. Thank you. Josana, do you want to present what you are doing in?
Yeah, thanks. So my name is Josanna van Dongen, and I attended also the Neuromodulation conference in New York, where I met some of you. Just a short introduction. We are just starting up our studies in my SAFER lab. It's a lab in which we investigate social, affective, and forensic electrophysiological research to promote a safer society. And in this case, we are investigating the usefulness of non-invasive brain modulation, for example, tDCS, to also increase empathic abilities, assuming that people who have alcohol or cocaine addiction have reduced social cognition. And we try to enhance these social cognitive abilities to also reduce aggressive behavior in forensic patients, addictive forensic patients. And that is actually our main interest here, using tDCS, but also EEG. So we monitor also what's happening in the brain after using non-invasive brain stimulation. Look at the connectivity within the brain using EEG. That's our main topic. Thanks so much.
Thank you. Any other person who wants to present or discuss about any specific question?
Well, maybe I can ask a question. Sorry, I missed the first part of your discussion. I noticed that a lot of people use tDCS or rTMS. Do you have a point of view as a group of how you would want to discuss these or compare the effects of these two techniques?
Okay, any idea, any comment from colleagues? The question is about do we have you compared the effect of tDCS compared to rTMS? What are the kind of evidence for that?
Yeah, so it's not a question like I know that there are differences, but it's more how do you as a working group want to integrate these two or address these two different techniques that might be beneficial for the treatment of addiction. But I was just interested if how you are using this.
I mean, this is Maron Bixen. I think it's an interesting point because these fields are sort of, they're in the same field, and a lot of people who do one tend to do the other, but they are distinct interventions at the same time.
Yeah.
And I also don't think, of course, it's not a race, you know, like, it's not like which one's better. I mean, until no one suffers from addiction, we don't really have to worry about that. And so I see these as sort of several horses. And of course, Flavia also mentioned tACS, and there's a broader family of techniques also that are in the same field. But again, you bring up a very interesting point about how do these things push up against each other?
Or this is Flavia, or we could rephrase the question, how can we study their synergy?
Yeah, I like the rephrasing. Yeah.
Of course, from a theoretical standpoint, a combination of the two techniques might be synergistic. I think maybe Michael can expand on this. He had some work showing how when you do some tDCS and then you add the TMS, you induce greater neuroplastic changes. But this would require lots of studies. I'm not aware of studies using the combination for treatment as a treatment strategy. Do you know of anything, Michael?
I think there were some very early trials where they tried to enhance efficiency of one by the other techniques, so some kinds of priming experiment, which were, however, as far as I remember, not extremely successful. The problem is that by combination, you make things also complicated. You can have synergistic effects, you can have also antagonistic effects. It depends on the specific stimulation protocols, the time interval between the protocols. I would also say what you're doing depends within and between techniques on the specific protocols, and the sense to use one or the other depends also on the specific intervention protocols. Yeah. So, for example, I would personally, for physiological reasons, also promote that online combination between stimulation and, for example, some therapeutic aspects. It's an interesting point. That's obviously that's tricky to do this with rTMS. So there might be, but I'm of course biased because I'm doing primarily tDCS. There might be a specific advantage of tDCS regarding these online things when you combine this with phytotherapy and find something. On the other hand, if you think about combinations of medications, we have different knowledge regarding the different stimulation techniques. So this might be also something pragmatically to take into consideration. If you talk about tACS, or if you broaden the parameter range, then we have different physiological effects more clearly, which might make sense to think about also this. If you want to alter oscillatory activity, if you want to use plasticity. There also maybe a combination of rTMS with tACS might make sense. I think it really depends on what kind of effects you want to get, not only therapeutic, but what's your physiological idea, what to change and how to change, and this might in some way determine what you're doing beyond pragmatically.
Great, thank you very much for everyone for attending this webinar. We, in terms of the next steps of kind of how we might be able to move forward, we are right now planning for the second INTAM webinar in something like 4 months, in mid of January, in January 16. So please mark your calendar for January 16. And meanwhile, we are moving forward to have a checklist of items that we want to kind of consider as quality measures, and also how we might be able to have shared outcome measures. So we are moving forward to have a consensus paper together. And besides the consensus paper, we are also sharing a checklist that we want to have people's idea about the checklist. So we are hopeful to be able to also have a quality control checklist together. So that would be the next steps that we try to do together. And meanwhile, there are upcoming meetings that I hope that we can meet each other in upcoming meetings. So just for making kind of a concluding remarks, those who we already have here, if there's any kind of concluding remark, that would be nice. Marom, Flavio, Shirley, I mean, all those people, Abraham, people that we have in the meeting, would you have any kind of concluding remark?
Maybe before we come to this, is 16th already fixed? Because then I'm exactly at this date, I'm in the plane from Tehran back to Dortmund. So should we maybe have to do that most of the people can attend, or?
Sure, we can definitely. What I will do, I will share the timing with everyone, and if there is any specific kind of conflict that people would like to change, we would definitely be flexible in terms of having everyone on board. So we just want to get sure that people would be on board. So we definitely will be about that, right?
Thanks.
This is Marom. I would just say, first of all, thank you, Hamed, for organizing it. And I just want to say I was so encouraged just by the tone of this discussion and just the professionalism, and I just feel very good about this community, and just based on this initial discussion and how it went so well, that we can continue to move ahead.
Thank you. Any other conclusions?
This is Flavio. I also would like to thank you, Hamed, for putting this together. I've noticed several things which I find very encouraging, and I just want to highlight one is the diversity of the people on this call. In many regards, I think that's how we can best do science together as a community. I'm looking forward to work with all of you. Thanks, Hamed, and thanks to all of you for being here today.
Any other concluding remarks?
Yeah, one more thing, maybe coordination between everyone is great. Also coordinations between societies, because I'm aware I'm involved already in two different societies dealing with brain stimulation and addiction. And I think it would be important maybe to try to make one meeting so that we don't have 5-10 meetings per year for each of these societies. I think there are maybe 3 already. So it would be maybe important for you, Hamid, as the organizer of this, to get in touch with the other communities and maybe try to have one addiction stimulation type of community instead of having too many. But this was a very nice discussion. I think it's a great initiative, and congratulations for being able to organize this.
Great, thank you. Michael, any concluding remark?
I think it was it's very nice to see little discussion, professional discussion. Don't have much to add to what Marom said. And I think it's an important initiative to somehow to coordinate efforts and to set standards. And I learned also a lot about new things are going on in the field today, which is nice. And should definitely go on. I'm looking very forward to the next meeting.
Great. Thank you very much, everyone, for attending the meeting. We are exactly on time, so hopefully you would have those who are in the middle of the night, they have a nice night, and those who are just starting their day, they have super nice day. So let's keep in touch. We will keep in touch by email, and we will meet each other soon. Have a nice day.
Have a nice day. Thank you.
Thank you, Matt. Bye-bye, everyone. Bye.
Bye.